Hi, my name is Niamh Kearney, and in this animation, I'll be explaining how cells communicate and repair a multicellular organism in the process of cell signaling and mitosis. Imagine a random person. A random person is enjoying a day, standing outside and living their life. Out of nowhere, a ball suddenly comes in and knocks them on the ground. They still remain conscious and thankfully didn't hit their head, but they happen to have a small cut on their hand. But no matter how small the cut, their body needs to be healed to stop the bleeding and infections or disease. Let's zoom in a bit on just how this process happens. Now, sadly, cells don't have space station communication rooms like in Star Wars, but your cells are constantly communicating with each other, with hormones, ligands, and/or signal molecules. With that in mind, imagine that we have this cell who's in G1 minding its business. It then receives a signal molecule from another cell that tells it to secrete some ligands. These ligands it has been told to produce will induce mitosis in order to repair the cellular damage from the cut. The cell then completes the three main components of cell signaling. Reception. A receptor in the cell membrane receives the signal molecule and causes a shape change in the receptor. Transduction. The shape change induces a signal transduction pathway that carries out the signal, or multiplies the signal. That's called amplification. Response. The activation of some kind of cellular activity, either in the cytoplasm or the nucleus. In this case, the response is in the nucleus in order to encode for the ligands by means of transcription and translation. Now, the encoded ligands have been secreted outside the cell and into the bloodstream. This is called endocrine signaling. But where do the molecules know where to go? They don't. As are most things in biology, reception is random interaction. The molecules don't have maps or genetic code for direction that tell them to go north through the bloodstream and take two lefts to the deltoid. The ligands will try to bind to any random receptors on random cells, but they have been encoded specifically so that they don't fit the shape or cause a shape change unless it's a specific receptor on the right cell. So the ligands go bumping around until eventually they reach the correct receptor, which is on the correct cell. There are three kinds of protein receptors: GPCRs, or G-protein coupled receptors, ion channel receptors, and RTKs, or receptor tyrosine kinases. Once the ligand binds to the RTK during reception, it causes a shape change. This is when the RTK dimerizes and autophosphorylates. Inactive relay proteins with phosphate from the RTK now become active relay proteins that transfer the signal to induce a transduction pathway called a phosphorylation cascade. And quick tip, when I think of phosphorylation cascade, I like to think of a waterfall. Waterfalls are cascades of water, so phosphorylation cascades are cascades of phosphates. In short, the active relay molecule phosphorylates the first protein kinase. This phosphate is then transferred to two other protein kinases, thus triggering an inactive protein to become active, which brings about the cellular response. This particular cellular response calls for the cell to do mitosis. Quick recap, mitosis has two stages. Interphase, the cell's preparation for mitosis. G1, the cell lives normal life and its job is being carried out. S, the synthesis of the DNA within the nucleus of the cell. And G2 is the replication of the organelles. And then there's the mitotic phase: prophase, metaphase, anaphase, telophase, and cytokinesis. All of which I think kind of sound like superpowers, but that's probably just me. Anyway, mitosis is a highly regulated process, and there are multiple checkpoints to make sure that the next stage is ready to be carried out. You wouldn't want to induce a cell to divide with unreplicated DNA. That would be very bad. And you wouldn't want to have a cell do mitosis unless necessary, which in that proportion can lead to cancer. But that doesn't happen because the activity of cyclin-dependent kinases, or CDKs, and MFP helps regulate this process by only being present in certain stages of the cell cycle. Now let's fast forward a bit. We have the cell in G2 and all the DNA and organelles are replicated, and the cell is ready to divide. We start with prophase. The duplicated DNA strands become condensed to sister chromatids, conjoined at a place called the centromere. At the same time, the structures of microtubules begin to form in the cytosol, stemming from the centrosome. The nuclear envelope then begins to dissolve, and some of the microtubules begin to attach at a place called the kinetochore, which is a specialized protein structure at the centromere of the sister chromatids. The other microtubules are beginning to interact with the microtubules on the opposite side of the cell. Next is metaphase. The chromosomes are aligned along the metaphase plate, which is like the diameter equator or prime meridian of the cell. The centrosome's kinetochores for each chromosome is perpendicular to the microtubules on the opposite poles. Then there's anaphase. The sister chromatids are pulled apart from their centromeres by a motor protein within the kinetochore. It breaks down the tubulin from the microtubules, drawing each sister to the opposite side of the cell. Then there's telophase. The non-kinetochore microtubules have stretched the membrane of the cell two nuclei begin to form in each daughter cell around the chromosomes, which begin to decondense, and the spindle microtubules are depolymerized or broken down. And finally, cytokinesis, which often takes place at the same time as telophase, but in the case of animal cells, the cell membrane cleaves inward on itself until eventually you are left with two identical daughter cells. This entire process of signaling and mitosis happens hundreds of millions of times over until eventually the cut is healed. Now just some summary. Some key vocabulary are, with cell signaling, reception, transduction, and response. Secretion, endocrine, paracrine, and autocrine. Receptors, GPCRs, ion channel receptors, and RTKs. There's also intracellular receptors. Mitosis is prophase, metaphase, anaphase, telophase, and cytokinesis, or PMAP. And there's some key concepts as well. Cells are in constant communication with themselves and other cells. Ligands can only fit in the specific receptors they are encoded for, and mitosis is a highly regulated process. And with all of that, I hope that this animation helped clear up any misconceptions or questions that you had, and good luck in AP Biology.